Welcome to the latest edition of the Mind-Gut Conversation, the interview series about topics related to health of the brain, the gut, and the environment. Today's guest is Dr. Elaine Hsiao, who is an assistant professor in the Department of Integrative Biology and Physiology at UCLA, and despite her young age, an internationally recognized authority on the interactions between the gut microbes, the immune system, and the brain. She has published seminal papers on the role of the gut microbiome in mouse models of autism spectrum disorders and epilepsy. She's also won numerous awards and has given a highly recommendable TED Talk on the topic altering microbes, how the microbiome affects brain and behavior. Dr. Hsiao, welcome to the program.
Thanks so much, Emeran. I'm really excited to participate.
Okay, so let's jump right in. You belong to a small group of basic scientists around the world who study the influence of the gut microbiome on the brain, and you have published some paradigm-shifting papers on the role of microbes on behavior in mouse models of autism spectrum disorders and epilepsy. What got you interested in this new field of neuroscience?
Yeah, so I was really lucky that early on I was always interested in environmental interactions with animals. And so my earliest research experiences were studying how environmental exposures can modify histones. And I was later on really interested in neuroimmune interactions with the nervous system. So the same idea where our environmental interactions with immune challenges can influence brain development and behavior. That was actually when I was a graduate student with Paul Patterson. And at the same time I was studying immune contributions to autism, I was noticing that in the microbiome field, really exciting work coming out on microbiomes and how you can use select microbes to modulate the immune system. And so that was the initial thought, that I draw from pioneering research in that area and apply it to the immune-mediated models I was studying for autism.
So in many ways, I mean, the field has sort of moved from the gut to the incorporating the environment as a very important factor. So you kind of, in your career, you sense that already early on that this is a crucial connection.
Yeah, I'm not sure that I really knew what I was getting into early on, but it was just, following where the science was taking me. When I started in the Patterson lab, these ideas were really, new, and they were considered fringe to the field. And so I'm not sure that I really had the foresight, but I just was interested in the question and was interested enough to continue working on it.
So one study that was sort of your first appearance in sort of the high-profile neuroscience literature was on the autism spectrum disorder mouse model and looking at the role of the microbes in that model. Could you summarize just in a couple of sentences the main findings of that study?
Yeah, sure. So in that study, we were studying a mouse model for autism that was immune mediated. And we found that in this same model that has the behavioral characteristics of autism, they also had immune problems and GI problems. And so we applied a bacterium called B. fragilis that in the literature was already known to treat symptoms of IBD. And we found that treating the animal model for autism with this microbiome-related intervention was enough to correct the GI problems and at the same time correct some of the behavioral problems. So it's a really early proof of concept that microbiome-related interventions in mice could have an effect on autism-related behaviors.
So there's obviously, this study has brought up many questions, and clinically these interactions of gastrointestinal symptoms, brain and behavioral symptoms, psychological comorbidities. It's a very complicated disease, obviously, and in the mouse, you were able to dissect some of these components, but it's it's obviously a real challenge to translate this into your findings into human patients with ASD. Do you believe that some of your findings are relevant for patients with this disorder?
I agree completely that, we barely, touched on some of the phenomena that we observed, and there's a lot still to be done in studying, even at the basic level in the single mouse model, what's going on. In terms of translation to human ASD, I really exercise caution in doing so. Just recognizing that a lot of the things that we find in animals may not translate very well. A lot of the animal models that we study are simply models and not really, they don't really encompass the entire complexity of the human disorders. That being said, the implications for the study, if we do make a big leap, the implications would be most relevant to people with autism that have comorbid GI problems. So it may not, autism being such a heterogeneous disorder, there may not be one intervention that will work for everybody. And for us, we were really studying a very specific subtype and root of autism. That has an immune component and a GI component.
So another question to this topic: how strong is the scientific evidence that alterations in the gut microbiome play a causative role in brain or in gut symptoms of patients with ASD, or as opposed to thinking that it's always the question of causality, that the brain alterations could cause many of the gut and microbiome changes that are being observed in both patients and your animal model?
Yeah, so I am very also cautious about using that word causative. I think right now there's very little evidence that microbiome changes are causal to autism overall. I think that it may be more likely that the microbiome may be a disease modifier, so that there's a whole host of other causes, but that changes to the microbiome that are drastic could modify the manifestations of the disorder, so symptoms of the disorder. That being said, on the basic science side, there are other labs that are kind of publishing newer data that can hit at this possible modification or even causation. Some of these studies are showing that transplantation of autism-related microbiomes into animals could cause some of its symptoms. Or they're also showing that things like fecal transplant could correspond with some improvements in autism symptoms. All of these are very kind of preliminary and early proof of concept studies at this point.
Yeah, I mean, I like the concept of the microbiome playing a modulatory and not necessarily a primary causative role, because I think in other brain disorders like depression, anxiety, it's sort of a similar story. I mean, there's now good evidence from mouse models and from mouse to human transplantation that there's definitely a role. There's absolutely no question. But I think something has happened early on in this field that the media got so excited based on the early animal studies that the expectations have risen of patients, particularly with something as devastating as autism spectrum disorder. And sort of people have forgotten this. There's a lot of neuroscience already existing, in many of these brain disorders, like decades of research that still is relevant. So I personally think what you said about this modulatory role is probably the best way to reconcile what we see in humans, what we already have known in humans, and what's now coming out new from the mouse models.
Absolutely, absolutely. At the end of the day, although there are all of these peripheral comorbidities, it is a behavioral disorder and would ultimately affect brain and circuits underlying behavior. And so I think studying the interactions across the board are really interesting, and not really trying to pinpoint just a single cause, that it's the microbiome only without any question of as to what's going on in the brain, and vice versa, I think is really important.
So one question that always intrigued me has to do with the role of maternal inflammation in increasing the risk of autism spectrum disorders. So the model that you use, the maternal immune activation model, uses an inflammatory stimulus in the pregnant mother. And there's lots of epidemiological studies that have demonstrated an increase in the disease during times of viral disease epidemics. So wouldn't this suggest that it's the maternal inflammation and not primarily the infant's microbiome that plays a primary role in the brain abnormalities?
Yeah, that's a really good question. In a mouse model like this, although it sounds very simple, you know, there's an immune challenge during pregnancy, then there's a whole cascade of events that happens at the molecular and cellular level of all of these changes to biology that could all contribute to how the offspring develop. And so the initial trigger here is maternal inflammation. The initial trigger is maternal inflammation, but downstream of that, the microbiome changes, brain development is also altered. At some point, the offspring also develop GI problems. And so it really is a trigger that leads to many different consequences. And downstream, each of those consequences can all contribute to behavior.
Okay, so another question related to this topic of maternal immune activation. So probably one of the most common forms of low-grade immune activation in humans today is diet-induced changes in the gut microbiome and intestinal permeability, resulting in a situation called metabolic toxemia. Is there any evidence that obesity and particularly the metabolic syndrome in pregnant women increases the risk of autism in the offspring?
There is some evidence that maternal obesity, maternal metabolic syndrome could be associated with abnormal development in the offspring. I think the link to autism is not quite as well established as compared to other environmental insults, but there's some evidence to suggest a connection.
Mm-hmm. Shifting to another publications, another paradigm shifting study on the interactions of certain gut microbiota with serotonin containing so-called enterochromaffin cells in the gut. As probably a lot of people know by now that more than 95% of our body's serotonin is located in these specialized cells in the gut, just microns away from the microbes. Could you briefly summarize the key findings of that study?
Yeah, sure. So this was a study also in mice where we were following up on a lot of existing metabolomic data. So existing data is already showing that if you raise mice with no microbiome, that they end up lacking a lot of gut serotonin. So about 50 to 60% of gut and actually blood serotonin as well. And so we followed up by studying whether there are select microbes involved and how exactly this may happen. And so we found the select group of microbes, they're mostly spore-forming microbes from Firmicutes, that seem to induce serotonin synthesis. So they send metabolites to these enterochromaffin cells and stimulate them to synthesize more serotonin. And so we found that downstream of that the serotonin that was microbially modulated is also important, so that it could have a role in activating neurons that underlie motility and for modulating hemostasis of blood platelets.
Does the serotonin that's sort of partially controlled by microbial metabolites, does that peripheral serotonin does it have any influence on brain function?
That's a great question. That remains an active area of study. I think historically it was thought that the peripheral serotonergic system is entirely separate from the brain serotonergic system, and that, you know, serotonin itself doesn't cross the blood-brain barrier very well, but maybe some of its precursors, tryptophan and 5-hydroxytryptophan, do. More recently, there's more and more interest in actually gut-to-brain signaling through sensory neurons. And the finding that these chromaffin cells may be able to innervate vagal neurons directly, and that things like serotonin are known activators of vagal neurons, so that you could potentially stimulate serotonin from the gut and affect signals that are transmitted into the brain. But that exact pathway altogether is really at this point piecemeal of different evidence combined, and it would be really interesting to follow up to see if it actually does happen.
Yeah, this remains an area all the way back from the time when these serotonin containing cells were first identified and characterized to until today, that we really don't understand why evolution puts, the great majority of this neurotransmitter that's involved in so many important functions like sleep and pain sensitivity and mood, food intake, why all this is sitting in the gut and is influenced by the microbes, but we don't really know if it has any influence or what type of influence it has on these brain networks that control these various functions and behaviors.
Exactly. I'm really excited about this area of research and just finding that there's a lot more to learn about what gut serotonin does.
So one thing that's also been, having been involved in the human side of these studies and, based on some of our scientific interactions and collaborations that we've done. So results from human studies on brain-gut microbiome interactions have lagged behind the exciting paradigm-shifting studies performed in mice. Since you have been involved in both these types of studies, the mouse studies in the laboratory and human studies on patient populations, what do you think is responsible for this lag?
Oh, that's a great question. I think what I learned, especially also from our interactions and learning from what your team does on the clinical side, is just how incredibly difficult it is to do a human study. How there are so many variables, especially with the microbiome, in that it responds to many different types of exposures, that there are just so many variables that need to be controlled for. And so one big challenge is really, having the bandwidth to do a very, very comprehensive and well-controlled human study that takes into account all of the variables like diet and, xenobiotic exposures of each person that could contribute to differences in microbiome. And so I think that's very difficult. Of course, it's also really difficult to do interventions in humans. I think when we do animal work, we have the privilege of being able to really go in and intervene with severe methods. So even the idea that we can raise animals as germ-free, or that we could treat them with lots and lots of antibiotics just because we want to deplete the microbiome, that's not something that could be done clinically, of course. And so it's hard to prove causality there.
Based in your own work and your close communication with others in this area, which brain disease do you think is the most likely, so the lowest hanging fruit that we will see paradigm shifting, either diagnostic or therapeutic interventions based on microbiome science?
Oh, that's a tough question. I think just based on the history of the field, some of the earliest studies were really on microbiome and multiple sclerosis. So brain disorders that are known to have an immune component. So MS or even responses to stroke. I think that may be a bit further along. It also leverages expertise in using microbiome interventions for purely immune and autoimmune disorders. So that may be maybe the furthest along, since the research was some of the earliest. Aside from that, I think there's a lot of interest in autism and Parkinson's disease, because subsets of them, of individuals with those disorders, do have GI problems. And so that may be a little bit more tractable as well. That you can use microbiome-based treatments for GI problems and to see what happens at the same time to some of the neurological symptoms. So yeah, it's hard to say. There's so much interest in all of these areas, but I think just based on history and practicality, those may be the first to see the light.
So you didn't mention another really fascinating study that your lab has done in a mouse model with of seizures, of epilepsy. And the interesting thing for me in this particular case is that there's already pretty good clinical evidence that dietary interventions in kids with refractory seizures can be helpful, particularly the ketogenic diet. And so this is sort of an area where we go into where there's already substantial human data that supports that there's something between diet and brain function. You want to say a few words about that study, which I think is really remarkable?
No, thanks so much for raising this. I mean, I think this is another case where it may be a little bit more tractable because it is already being used as an intervention. So the ketogenic diet is a very high fat, low carb diet that's already integrated clinically for treating kids with refractory epilepsy. So that's kids that usually after they've failed their third treatment with an anti-epileptic drug. And so, yes, we were finding that the ketogenic diet, again in mice, changes the gut microbiome dramatically, and that the microbiome changes are important for mediating the anti-seizure effects of the diet. And so the implications are whether we could find microbiome-based interventions that either promote the efficacy of the ketogenic diet, or even, one day replace the ketogenic diet for treating seizures.
I mean, this is a really good example. I'm personally not a fan of the ketogenic diet, which is obviously, there's a whole, it's almost like a religious movement behind that. But it's interesting that there's certain disease areas, including, the seizure disorders, where the basic principles of that diet really can be used as a therapy, just like we use antibiotics as a therapy, but obviously don't recommend people staying on antibiotics as a regular feature of their daily diet.
Absolutely. I mean, I think the same goes with the ketogenic diet. Although it can really help with seizures, one of the main reasons for discontinuation of the diet is just all of the side effects that it comes with. And so even though, it's a priority list of what you want to target, I think if one does have epilepsy, it would make sense to want to treat the seizures, and it may make sense to have to deal with the side effects in doing so. So yeah, we hope that learning more, especially about the microbiome component, and maybe we can learn more about how to control side effects of the diet as well.
So we already talked a little bit about this phenomenon. So some people, talk about that this field of brain-gut microbiome interactions has created some sort of a bubble, mainly, and the people responsible to a large degree, the media, who have amplified the message and extrapolated to breakthrough therapies for many intractable diseases. What do you think the science in this area will be in 10 years from now? Do you think that there will be a few areas that will come out as really what the science completely changes our paradigm, diagnostic tests, treatment approaches, whereas others may just fall by the wayside and people say, well, it was just an epiphenomenon, the microbial changes?
Yeah, so I think on the microbiome side, I hope that where the science will be in 10 years from now is a much deeper understanding of mechanistic interactions between the microbiome and just health in general. So, of course, you know that with the microbiome field, it was very new. That it's only in the past decade or so that we've really appreciated all the types of microbes that are indigenous to the body. And there's a lot to do in just studying fundamentally what do they do, what types of metabolites do they produce, how do they modulate the immune system, and how do they interact with neurons and other cell types. And so I hope from 10 years from now, we'll have a much deeper understanding of these intersections between microbiomes and the nervous system. In terms of application to clinical work, I think that I hope that 10 years from now, there will be also more attention to microbiome, just kind of the role of microbiome changes to health and disease. I think right now there are very few examples of where microbiome changes actually translate to clinical outcomes, that there's really strong evidence for that. So I hope there'll be more clinical evidence to either test whether microbiome changes do contribute to chronic disorders, or to disprove this idea that certain microbiome changes don't really matter for particular disorders.
Yeah, so my personal impression or thoughts about this topic, whenever I think about the evolutionary history, how the microbes of the oceans first settled in these first animals, marine animals, and developed this interaction with nerve cells in these floating digestive tubes, and then used the same neurotransmitters, the same signaling molecules. Whenever we look at that that fact that over billions of years, you know, there was this close, well, I mean, first the communication between the microbes themselves, but then the close communication with the nervous system of these primitive animals, and then the same neurotransmitters then pop up in the brain. Whenever I think about this, you have to say there has to be a very important link between the brain and brain diseases and the microbes. It's just undeniable. And to find that out, what this exactly is, I think will be the main challenge.
Absolutely. I think it's just so amazing to realize that so many of these systems are so integrated. So the fact that the microbiome seems at the interface of our interactions with the environment, it responds very quickly to environmental changes, and that some of these changes can be, or some of the responses in the microbiome can persist for longer than the environmental challenge does. And also that there's so many intersections of the microbiome and the immune system and the nervous system. I think these three systems in particular are common in that they're dedicated, you know, sensory and almost surveillance systems as well. So that each of them, the immune system, nervous system, and the microbiome, responds very quickly to the environment and interact with each other.
Okay, well, let me end the conversation on a sort of personal note. So you've had a remarkable career from basically your name was not known, you know, 5-10 years ago, to becoming a rock star in this type of in this area. What recommendations would you give to young scientists that you know that want to embark in this area, that would help them in their career and sort of create a similar trajectory?
Well, thank you for the kind words. I definitely don't feel that way as an assistant professor here. But yes, I mean, I think I had a great privilege of being able to do science in an environment that was very supportive, and that gave me a lot of freedom. And so my advice, or the way that I got to my point in my career, this point in my career, was really just having the liberty to study wherever the science would take me. You know, there'd be interesting questions that would come up that they may not be the main foci of the labs that I was in, but that my mentors allowed me to study them. And so I remember when I was in Paul Patterson's lab, my earliest interests, I joined the lab to study neurological bases of autism and became really interested in brain development, but then somehow became really interested in placental development as well. And so it was a little bit tangential, but I got to really study that. And when I got interested in the microbiome, I also was fortunate to be able to just pursue wherever my interests were taking me. And so I think that for the microbiome field, that there's a lot to be studied, and it can take you anywhere, but as long as you study the questions in a very rigorous fashion, I think that it's worth doing. So that's my, that's my advice.
Well, thanks, Elaine. It was a great conversation. I hope the viewers will enjoy this as much as I did, and look forward to your next papers, which I'm sure will be, again, touch on paradigm shifting topics. Thank you very much.
Thank you so much.